Thank you for coming for um, the seminar by Steven Rothstein. It's my pleasure to introduce Dr. Steven Rothstein from the University of Guelph. Steven is a professor and senior research chair for the University of Guelph Molecular Biology Department. I've known Steven for, I don't know, since 1990 or something. Steven has had an extensive um, a range of experience from very basic fundamental science all the way to the ultimate field research. Steven did his PhD in biochemistry at the University of Madison, working on transposable elements and bacterial transposable elements involved in antibiotic resistance. And then he went on to work for his postdoc in Plant Breeding Institute in the UK. And then he came out and immediately joined the Ciba-Geigy, which is now called, I think that's Syngenta, part of Syngenta. And there he was one of those pioneering scientists who started out with some of really elegant technologies that have been used quite a bit in fundamental sciences, in plant biochemistry or any biochemistry. It involved expressing a plant protein, a secreted plant protein, properly folded in yeast and functionally expressed. The RNA, antisense RNA technology for downregulating genes. He was one of those pioneering people for that. And then once coming into pioneer hybrid, so from Ciba, he went to University of Guelph and headed a lab and was the head of the department of the molecular biology in University of Guelph. And from the University of Guelph, he went to Pioneer. And Steven was one of those early people who recognized that in order to be able to have a really good assessment of the changes that you're making in breeding, you need to be able to measure it. So Steven's instigation was the reason why Pioneer Hybrid built its first drought house and the first phenotyping facility in Pioneer as early as, well, I won't say that. And then it was also Steven's instigation that led to the starting establishment of managed stress locations, primarily for transgenic drought testing at that time. And Steven and Greg Edmeades were those people who actually set up that the whole scheme well before Bill Niebuhr and Mark Cooper and all of those people came on board. So like I said, Steven's experience ranges from the fundamental science all the way to the field, practical field. And I won't take too much time, but I just still need to explain to go through one particular experience I had. Now I'm from Steven's lab as a postdoc fellow there. And one of the projects that, so the lab had a variety of projects at that time, extensive. From one end, the guy behind me, Richard, was making spider silk, which has tensile strength, spider silk protein with heavy tensile strength. There were some people working on male sterility through self-incompatibility. I was working on a protein called hydroperoxide lyase that is involved in the defense mechanism, but also in the flavor profile of leaves, fruits, etc. So we first cloned the hydroperoxide lyase from tomato in those early days, and it was not very clear if that was hydroperoxide lyase or not. So we needed to do a functional testing. So we didn't know how to be doing that, but one thing, the simple test that we immediately did after purifying the protein, expressing the protein and purifying it, was to take it to a small test tube and adding the protein, purified protein, into the test tube and then adding its substrate. And lo and behold, the whole tube, entirely, you could smell cut grass in the test tube. And that was one of the first, for a biochemist, that is the most simple but most really rewarding minute where you can actually assign functionality to a protein that can then be used, or a pathway that can then be modified for something else. So Steven's lab has been working for a long time on nitrogen use efficiency. The regulation of nitrate assimilation was something that I did quite a bit in Steven's lab too. So that is the one topic that Steven's going to be talking about today, nitrogen use efficiency. So it was after Steven's time that Pioneer actually started depleted nitrogen locations for our nitrogen use efficiency trials. That was also modeled on the drought testing managed stress environments that we have now extensively for Pioneer. So Steven's gone back from Pioneer to University of Guelph, and that's where he is coming to us from, and he is now doing a brief sabbatical with us. So with that, it's a little bit of a long introduction, but I wanted to impress upon you the breadth of thinking that needs to go into functional, practical implementation of what we need to have in productivity enhancements. Thank you.
Okay, thank you very much, Shobha, and thank you for inviting me to be here, and to all of you who I've talked to for being great hosts and having very interesting conversations. So I sort of am going to talk about this topic today, not completely, but it is a topic that we've worked on for quite a few years. And before I do that, though, I want to give my brief capsule history of crop improvement and biotechnology, each one of which will take about 3 minutes. So we all know that with regard to agriculture, there are certain long-term pressures, growing population, changes in diet, increasing stress on the environment, particularly due to climate change and degradation of land. But nevertheless, over the years, and this, I'm using corn much throughout the thing, that's the crop, the target crop of almost all the work that we've ever done. And so what you can see here, and you could do the same with other crop plants, is that the yield over the years has gone up, and the only reason that the world population can go up like that is that the grain yields have gone up as well. But I just wanted to show a brief history of what happened and how this was done. And for the 1930s, I'm getting slightly old in the tooth, but I wasn't around in those years. But this is a slide set that I got from Don Duvik when I worked at Pioneer, and he was a former research head at Pioneer and a really great all-around guy who passed away a few years ago. And he had a slide set of what they used to do back in the 1930s for improving yields. And so the first breeders were not trained in plant breeding. They weren't trained in genetics. They knew nothing about genetics, really. The technology was pretty simple. This was the technology for planting. This is the technology for weed control. And for harvesting. Very, 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 very simple tools. Now, if we go to what happens these days, what you can see is farm technology is amazing. First time in my life when I was in Australia recently, I went in a combine. They're amazing machines. Chemical treatments of seeds, biotechnology, I should say tens and tens of millions of DNA marker points. And so what you can say in my little 2-minute spiel is to achieve similar yield increases today as you did in the 1930s, it obviously costs much, much, much more and involves many, many complex technologies, much more than it did in the old days. And Shobha mentioned that I worked at Ciba-Geigy during the first few years of the sort of what we thought was going to be the biotechnology revolution. And in that time, we did develop what we are now calling first-generation traits. So I'm just going to give you, as I said, my 5-minute history of developing the first generation of these traits. And the really interesting thing about working in biotechnology in those days was, boy, we sure did a lot of blue sky thinking. We thought we could do pretty much anything using biotechnology. But at the same time, we're missing virtually every single type of important capability one needed to actually do that. And we were more than a little bit naive about how easy or difficult it would be to do that. And I just wanted to demonstrate some of the things that we were thinking about. So, we thought we could develop the perfect nutritional food like deep-fried Twinkies. But in reality, how you can develop a food like you're still trying to do now, that has all the vitamins and the minerals and the other nutrients that you need. The first genetic engineered crop, I'm not sure how many of you remember this, but it was the Flavor Saver tomato from Calgene. And the idea was that you would develop a tomato fruit that would last better on the store shelves, and this was actually sold for a few years. And you could pick when it was more mature, so theoretically it could taste better. But the unfortunate thing is at least the variety they ended up developing tastes much, much worse than a regular tomato, so it didn't really work very well as a commercial product, and it sort of disappeared from the store shelves. One of the other classic things was a company out in California, I forget, Advanced Genetic Systems. I almost went to work for them, actually, Advanced Genetic Systems. And their big thing was they were going to produce bacteria, an ice-minus bacteria. So the reason that a lot of fruits and vegetables, if the temperature gets down to a fairly low temperature, the freezing is due to the fact that you have to have a crystal factor, and these bacteria were the things that would cause crystallization and frost damage. So the idea was you would go out there and spray, I guess this was strawberries if I remember correctly, strawberries with this bacteria that didn't have this, this ice nucleation factor, and this would prevent frost damage. As you can guess, this didn't turn into a commercial product. I do like their spacesuits they use for doing it. One of my favorite ideas was by one of my colleagues who I'll leave unnamed, a very senior person at Steve Geigy when I was there. And his idea was that all you really need to do to make insect-resistant plants is to find antibodies that are can bind either the insect mouth parts or the insect foot pads. You produce them in plants, and the insect would stick there, and that would be it. As you can guess, we're a little off on scale there. The insect mouth is quite a bit bigger than the antibody, so that didn't go anywhere. But it can show you the sort of range of things people were thinking about. Some ideas were crazy, some ideas not so crazy. So if you looked at, we actually started a nitrogen use efficiency in corn project way back in 1986. And how we were going to do this, what did we know? Not much. We knew that, I'm not doing the pointer very well, but that's okay. We knew that nitrate assimilation had 3 genes at that time. There was nitrate reductase, nitrite reductase, and glutamine synthetase, taken from nitrate to glutamine, and that's basically all we knew. We didn't know about the regulatory mechanisms behind it. We didn't know about all the other genes involved with uptake or downstream use of glutamine, etc. And we also couldn't transform corn in those days. All we could do is transform tobacco, but we still started a project. It was that sort of time. But even with all that sort of little, sometimes craziness and sometimes not, there were some important long-lasting projects that were done both by us and by other companies at that time. There was BT for insect resistance, herbicide resistance. We learned how to, as Shobha mentioned, modulate transgenic gene expression, which was not something we knew how to do before that. And at that time, we did also learn how to transform important crops. So it was around that time that we learned how to transform corn, for example. Okay, I just want to mention just one little bit about the effect of the first generation traits on yield. And this is taken from a paper, Shi et al., Nature Biotechnology. I haven't really looked up to see if anybody else has published things in this sort of area. But up to that point, it was really the only article I could ever find on it, and it really was not very perfectly well done. It's not, I wouldn't take it as wrote what they said. But basically, what this is the level of increase of yield based on different factors. Oh, sorry, I do have the pointer. So this is 1990 yield. Okay, so it's up to this point, and then The yield increase due to genetics plus management between 1990 to 2010 goes up to here, so that's in the conventional. And then BT corn borer adds this much, that little whatever that amount is there. So it's just a little bit on top of the thing. Now, I don't want to emphasize this too much because there's other reasons why BT can be useful. According to their data, if you add both BT and corn rootworm, you actually got no increased yield over conventional, although I don't necessarily believe that. So what are the advantages of the first generation traits and what can we learn from them if you're interested in doing new things? So, obviously the glyphosate resistance has been hugely useful. There's decreased herbicide costs. This varies depending on crops. Some crops it hasn't actually been that big a decrease. But glyphosate is much, much easier to use, and you can do it at any time, so it allows for conservation tilling, which has been really important in developing increased drought tolerance. There's some decreased insecticide use. There's certainly decreased year-to-year variation, and that's simply due to the fact that the insect pressure varies a lot from year to year, and so you don't have to worry about that. I mentioned here the ease of use of glyphosate. It's not quite as useful now with some herbicide-resistant weeds, so you have to add additional herbicides along with it, but still, it's still pretty useful. And then the downside, of course, for the farmer is it's also significantly more expensive seed. But nevertheless, it's been incredibly popular with the customers, as you all know, and almost all corn is biotech corn now, just because of the insurance policy of the insect resistance genes and the ease of use of the glyphosate resistance. But there does raise a really important issue that I think has been a killer with regard to trying to develop other types of more complex traits. So we're talking about traits like drought tolerance, nitrogen use efficiency, yield, and that is that if BT only has X amount of difference, can we find a BT equivalent additional gene, and what do we need to do in order to detect that? And the problem is that it's what I call the tyranny of small differences. You can sort of, from the Shi et al. Data, which again is not perfect, so I don't want to take it as rote, but you can calculate how much you would need to do in order to see statistically valid differences, and it's really a lot of cost and effort in order to do that. So 6.5 bushels per acre in corn, that's about a 3.5% difference. So that's a big difference. If you could do that, that would seem really good. You would need, at a 0.05 thing, you need about 260 plots in order to see that. May not sound so huge, but the problem is that you need to do that for every single construct and within a construct, every event in the construct in order to do it. So it gets to be a very expensive process, something that no one really wants to do. And that's probably the biggest problem that we face in trying to do those sorts of things. I'd like to use a few slides that I used to teach genetics, and just to exemplify what I mean by this. So in this example, it's a Great Dane here dog and a Yorkshire Terrier, and this is an example where different genotypes give different phenotypes. Okay, it doesn't matter how much a Yorkshire Terrier eats; it's never going to be as big as a Great Dane. In contrast, probably in India you don't do pumpkin growing contests, but this is something that in the U.S. Fair number of rural communities do this. So who can grow the biggest pumpkin? And so here, genotypes matter, but even more important is the different environmental conditions you use. So they use lots and lots of water, lots and lots of fertilizer, and also they trim it back so that it's only one pumpkin per vine in order to get this really, really big pumpkin. So in contrast, of course, what we have to deal with is this sort of thing. You just don't see the differences nearly so easily. And so I'm going to come back to this later, how to assess the effect of genotype on phenotype given different environmental conditions. Chopin mentioned that when I was at Pioneer, one of the things that really bothered me when I first got there is people would be doing assessment of drought tolerance, and we had no way of measuring that. So they could put things out in the field, but you could never get data that would say it worked, it didn't work, or you couldn't tell one way or the other. And so I think it's incredibly important point that you need to have the ability to measure the conditions under the condition that you want. Okay, I'm going to talk a little bit about this whole nutrient efficient traits and why occasionally companies are interested. So I say occasionally, it goes up and down in interest. Right now it's really down, but you never know, it may come back again. So the reason is obvious that it's N-based fertilizer costs are 80 to 100 billion annually. If you could decrease the cost, theoretically you could try and capture a portion of that if you have a solution for it. The reason the companies go up and down is it's hard to know how to capture that portion. But anyways, I won't go into that today. The other thing that's really important is just the level of increase in the amount of fertilizer used and the problems that this can cause from an environmental point of view. So there's a value thing here in dollars, but we also have a major problem in the sense that we're fertilizing the nitrogen cycle in the world. And I'm not going to go into this in detail, but all of the sort of natural sort of things are small compared to what humans are doing. And a big part of what we're doing is adding 100 million tons of fertilizer to the world. And this causes major environmental problems. And to give you a notion of how much nitrogen fertilizer is wasted, so this is the part not used by crops, I did sort of a calculation. So this is the yearly flow of Niagara Falls, which is not too far from where we live. And so this is the maximal flow because sometimes they decrease it for energy generation. And you wanted to, and you calculate the amount of nitrogen by saying what's the amount of nitrogen that gives you maximal growth of most crop plants, and that's in solution. And so the amount of water that flows over Niagara Falls in a year is about equivalent to the amount of waste nitrogen that's put out in the world. So that's an awful lot if you can think about how much that is. Okay, so let's just talk a little bit about nitrogen assimilation and use. And so this is just a diagram I got from my good friend and colleague Alan Good at the University of Alberta. There's a certain amount of available nitrogen in the soil. It comes from a few different sources. The main source in a crop like corn is fertilizer nitrogen, but of course in other crops it can be fixed nitrogen. There's some from precipitation. And then there's a certain amount lost, a certain amount is lost, gaseous losses into the air, leaching into the groundwater, and some of it's just unavailable in the soil. And then the rest of it is what the plants use. And you can sort of divide what it uses into two categories. There's the recovery efficiency, so how much is taken up essentially by the plant and maintained in the plant. And the other, so that's recovery efficiency, and the other is physiological efficiency. What portion of this taken up and ends up in the grain? So you can think about it in this way, but from a practical point of view, we really didn't care one way or the other which part of this process we were working in. So we took the approach that we were going to let the plants tell us which things made sense and which things didn't make sense, rather than start with a particular gene type. Okay, so the overall question that we had and we continue to have, we in the greater community, not just me, is can we improve essentially on randomness for genetic improvement for important complex traits? So we know breeding selection works, but can we use our knowledge in order to improve on that? So this can be for either breeding selection, I'm not going to talk about that today, selecting genes for trait improvement for transgenics, and how do we utilize all the potentially available data in a practical way. With regard to nitrogen use efficiency, then the question just simply becomes, can we use gene knowledge to maintain yield while decreasing the rate of N fertilization, or alternatively increase the yield under the same N? And this one's cheating a bit because that's happening naturally anyways. The amount of fertilizer used for corn is not going up while the yields are going up. but either one has the effect of increasing nitrogen use efficiency. Okay, so the whole idea was decreasing cost to the farmer and then decrease environmental cost. And the problem is, of course, you need to develop highly predictive tools and low-cost screening methods to be successful for this type of trait. And I can't say we've necessarily solved that problem. Okay, so in this work, we did this with a long-term collaboration with researchers from Syngenta. It lasted for about 11 years, from 2003 to 2014, and it was a really good collaboration. It's unfortunately ended now as the economics are not so good in some of these companies. But anyways, the idea really was you collect knowledge from a variety of things, just the obvious things that everyone does: genetics, genomics, transcriptome data, etc., model plants. And you use those for what they, at those days, called gene discovery, and then you would take those genes and test them in crop plants. I'm going to give 3 examples, I think 3 today. It might be 4, I can't remember. But one of them comes from completely from model plants, namely Arabidopsis, a set of transcription factors, and the others come from transcriptome data that I'll describe in just a second. Okay, so the approach using Arabidopsis is exactly the same approach we took on the crop plants of choice. And that is, we needed to develop defined and growth conditions. So this is obviously not in the field, but in defined growth environments. And then we'd identify as many mutants as we felt we could screen using the growth system, and identify the end responsive genes in this sort of way. So one of the genes that we found when we did this screen was this one that we called GNC for, what was it called GNC for? I forget, something nitrogen carbon anyways. I forget what the G was. And it turned out that this gene was important for modulating chlorophyll synthesis and regulating carbon metabolism. And so it sits, it's not an on or off switch, but it sits at a nexus of a variety of different pathways, and simply modulates the amount of essentially chloroplast and consequently chlorophyll that are present in the plant. And so what you see, the mutant has a very simple phenotype. It's reduced chlorophyll level in the GNC mutant. And if you take this same gene and overexpress it in rice, you see an increased level of green. Decreased level, you see a decreased level of green. So very simple. We did the same thing in corn. In corn, what you see is a delayed senescence in transgenic corn overexpressing this gene. You see an increase in biomass formation in these. We had a little bit of a problem. If you use a very strong promoter, it also affects the reproductive tissue, which is not so good. So you had to change promoters in order to do that. So this is an example. We took a transcription factor from Arabidopsis, we transformed it into rice and later into corn, and we see essentially the same phenotype. This doesn't always happen, I should say. As a matter of fact, it often doesn't happen. Whoops. So one of the things that we do typically do, and sorry, this thing is all messed up in the slide, so it's not going to be easy to see. But one of the things we do like to use transcriptome data for is to look at either a mutant versus a wild type or an overexpressing line versus a wild type in order to try and get an indication of what that particular mutation does. And so when you overexpress this rice GNC equivalent, what you see is an increase in expression of all the nitrate assimilative genes, a few other genes involved with nitrogen assimilation. You also, of course, see a change in chlorophyll level and some of the genes involved with chlorophyll biosynthesis. Okay, so that's the first type of approach that we use. And I'll come back to that particular one near the end of the talk. The second approach we did was to identify genes using profiling experiments. And this was done fairly early on in the collaboration at a time probably back around 2003 or so. And at that time, Syngenta had the ability to profile rice using Affymetrix chips. You couldn't do that in the public sector at that time. And so we designed a fairly simple experiment. And I'll describe the experiment in just a minute, but we identified candidate genes using this approach, and we chose 50 genes to test. And Syngenta researchers made transgenic lines overexpressing each one of these, and then we did the phenotypic analysis of all these. Okay, so the first thing, again, you need to do is, again, we defined a growth condition for rice, and what we wanted in this case was to define a condition where, in this case, 10 millimolar is maximal growth, 3 millimolar gives about a 30% decrease in biomass formation, but you still get some some rice grains formed, and 1 millimolar doesn't do so well, so it's about a 50 or 60% decrease in biomass formation. And the microarray design was really very simple. We didn't do anything fancy. In those days, we couldn't do that many chips. So we just did stable, high, medium, and low nitrogen. We did some transient conditions and induction and reduction conditions for 2 hours, and we collected tissue from shoots and roots. And we saw a certain number of significant differences, and this is just an example of what we saw. The numbers don't matter. You always see lots of changes when you do this sort of experiment. And then we chose the 50 genes based on that, and I guess I don't have a slide on the types of genes we chose. But basically what we did, you have a lot of them, and so you have to make some decisions. You don't tend to choose ones. You have no idea what the gene codes for. But we chose a few that were interesting just based on the amount of changes in expression in a couple conditions. So one of those was this gene, an early nodulin gene. So what you mean by that is during nodulation in nodule-forming organisms like soybeans, they've done the expression of sets of genes, and they found a set of genes that are expressed during early nodulation, and this rice gene is homologous to one of those genes, so that's all it means. We had no idea what the function of this was. We never would have chosen this as a candidate based on any criterion because we didn't know at all what it does. We still really don't know what it does. We have a little bit better idea, although I'm not going to talk about that today. But what we did know is it responded to both the nitrogen reduction and induction conditions. And so if you decrease, you see a huge change in expression, and if you increase the nitrogen, you see a huge change in expression. And if the conditions are the same, you see very little difference. And it turned out the overexpression of this was really turned out to be the best candidate that we found. What you find is that under the lower nitrogen condition, the overexpression gene had somewhere between 10 and 20% increase in seed yield. And even at the rich condition, it had about a 10% increase in seed yield. Now remember, of course, this is done under a very controlled condition, okay? And I wish I could tell you a lot more about this, but I can't. And I won't go into why it was never tested properly in the field, but just I'll leave it that based on politics in Syngenta and where they were going to do things, it never was actually tested properly. So we were left with that sort of unfortunate result. What we know about it, it's localized to mitochondrial membranes, and the overexpression of this gene in the same sort of way increases the expression of a certain number of other genes. So we overexpress the ENOD here, and what you see is an increased uptake of various forms of nitrogen. You see an increase in expression of GS1, a few other similitular genes, and you see an increase in amino acid flux. That's basically what we know right now, although we're doing more now with this gene in Arabidopsis, and hopefully we'll have more about that within the next year. Okay, so what can we say? Overexpression of ENOD93 increased rice growth and yield, more specifically under low-end conditions. And we don't know much about what it does, but it might play an important role in the transport of amino acids from shoot roots to shoots. Okay, the third example I want to give is another nitrogen responsive gene. And this one, under the different nitrogen conditions, we didn't see any difference between wild type. And I should say, we did 50 genes. Of those, 45 we didn't see any difference, right? So you mostly get negative results when you do that sort of experiment. But in this case, one of the people in the lab, Ashraf Al-Karimi, noticed that this particular gene was a MIB transcription factor, MIB55, had a very interesting promoter structure. It had a whole bunch of heat shock elements, response elements to various hormone, methyljasminate, ABRE for ABA responsive elements. And so he thought, well, maybe this has something to do with stress. And so he did the experiment. Most of the time, when we don't see anything under normal conditions, no one's willing to go back and try a different condition, to be completely honest. But he decided to do it in this case. And what he found was its expression is upregulated by high temperature in rice. So this is just using the promoter from the gene driving the GUS gene. So you see an increased expression at high temperature. And the growth of the MIB55 overexpression lines under high temperature we found was improved over that we saw for the wild type. And so this is maybe not totally clear, but what this is a change in dry biomass of the wild type, so the reduction percent of wild type versus those of the overexpression lines. And what you can see, if you look at dry biomass, the wild type is decreased by 55%, while the transgenic lines are only decreased by about 25%, etc. Same for the other measurements made. We did the same in maize plants, and we saw a very similar thing, but in this case we saw no change in drought tolerance in the rice lines, I should say. But in the corn transgenics, we did see both heat and some drought tolerance in the corn lines overexpressing this particular gene. Okay, so in summary about the OS MIB55, I'm not going to go through all the data. The overexpression lines show significant less reduction of biomass, height, and leaf damage and chlorophyll than wild type under heat conditions. Under water deficit conditions, the maize line showed the differences compared to wild type, while not in rice. And if you looked at the transcriptome of the MIB55 overexpression lines, you saw upregulation of a bunch of genes involved in the general stress responses, including heat shock and drought genes. And so that's all well and good, but as you know, or maybe you don't know, a lot of stuff you do under these defined conditions don't really play well when you put them out in the field. Oh, sorry, I have one last one to describe for you. Just because it's another way of thinking about the problem. We had one gene called another GATA transcription factor. It's a GATA12, whatever that means. And this is work by Guan and Lu in rice. And again, it was the same sort of thing that we'd seen before. This was a gene selected based on its nitrogen response, but we didn't exactly get a growth response that seemed very good, okay? So this is just the expression of the gene. It's expressed in a variety of tissues, more in leaf and root, and the same you can see for the GUS lines. And what you can see is the overexpression lines make a lot more of the gene transcript, and the RNAi make less, as you'd predict, so that's all nice. And what you see is not necessarily what you really want. So what you'll find is panicle length is the same, and the number of grains per panicle is the same, but the yield per plant is way down. Okay, so that's not a really positive sort of thing. The reason it's way down is total seeds are down, is that the dry weight is down, and what you also see, the number, I guess it's not listed here. Anyways, the number of tillers and panicles were down as well, so it's not on this slide. I apologize. So this doesn't sound such a good sort of increase in yield type phenotype. But one of the really interesting things was that if you grow this under high density, what you see for the wild type is that you get a yield per area that goes up a bit under, I'm sorry, I apologize. This is high density, this is low density. So what you see for the wild type is you get a decrease in yield under very high density, which is not unexpected. That's what you generally see. The really nice thing is in this particular line, if you grow it under high density, what you see, it has not a very good yield under low density, but under high density, you get an increased yield, and it's even better than the wild type under the lower density. And this density, by the way, is roughly what you do planting in a rice field, and this is obviously much higher than that. So this is just an alternative way of thinking about increasing yields, and it's actually the way many crop plants, of course, have their yield increased over the years by traditional breeding, by just increasing the plant density. Okay, so the overexpression of this gene resulted in a pleiotropic phenotype. I didn't talk about this. The RNA line showed an early senescence and less chlorophyll. And at low density, yield is much lower. Under high density, yield higher than the wild type. So that could be maybe interesting, maybe not. Who knows? Okay, the overall results from the rice lines, 5 out of 50 showed some positive change in phenotype. 3 out of 50, interesting from a basic point of view, meaning they looked funny. In some cases, you need to try different promoters to optimize the phenotypic effect and avoid negative pleiotropy. So that was true of almost all the cases that showed some positive effects. So only one of them that was not necessarily true. And this is the really unfortunate thing for us. The field testing had to be done by Syngenta. So why is that? We were capable of doing it, but for regulatory control and their sense of who should do what, they absolutely forbade us from doing any field testing of the corn lines that were generated. I should say, I didn't even mention this. For corn, rice, I said there were 50 different genes tested. In corn, there were over 100. So there was a lot of material there. Why was that unfortunate? Because within the Syngenta organization, sometimes they were willing to do this sort of thing, and sometimes they didn't. So there was a real stop-start sort of thing to this whole experimental system. Okay, so we had all these lines, and so if you were going to test them all, and they started to do it near the end of the collaboration, which unfortunately is not going on anymore, one needs to test on a large number of field plots, and you need low-cost, accurate, and predictive tests. And it's not trivial to do this by any means. Okay, so the approach that they decided to try and use was aerial imaging, essentially flying a plane over the fields, and using various imaging technologies. Probably most of you know what some of these are, at least. Spectral reflectance, both visible and infrared, thermal imaging, that's to help measure stress tolerance, LiDAR to measure plant height, so that's light plus radar for plant height, and then we were going to compare it to our ground measurements of the same lines. And so this was the field from our field from the plane. These are the color tests for the various imaging things, and these are the different corn plots that were out there. Okay, so there were a couple that looked really interesting from this initial experiment. I apologize, I should have changed this. This is this MIB55 gene that I mentioned. It's not just a transcription factor. I can say what it is now. It's the MIB55 gene. And what we saw both from ground imaging under that year in Minnesota, this was done in Minnesota, there was very, very little plant stress. It was a cool year, lots of rain. And even so, we saw some improvement with the imaging using NDVI of how the plants were growing compared to the wild type. And then the second very obvious one was this GNC gene. In this case, it was a change promoter. It was one that wasn't expressed throughout the plant. It was a Syngenta promoter that was just supposed to be in green tissue. And what you could obviously see was that the chlorophyll level was higher in the GNC plant. This is late in the growing season, so the idea is you can maintain photosynthetic capacity later into the growing season, sort of a stay green type phenotype. And at the same time, the senescence score of the wild type was higher than that of the GNC, so very obviously a difference. So the conclusions and issues with this, the transgenics of interest, actually, we were really happy that it did largely replicate the controlled growth results, little that they did with this. But, you know, to be completely honest, you need much, much more work to see if truly of any commercial interest. And what that means is for corn, you need to put it into test many, many pollen donors. So you use this as the female line. The transgenic is the female line. You use a variety of pollen donors. You need to do it on a wide number of field plots to see if it's work. And that's reliant on the company interest being sustained, which, as I mentioned, has not really been the case for financial reasons there. So you need sustained long-term interest, which is difficult for difficult traits like this. And I think it's the same sort of thing that we see in the public sector, where it's very difficult to maintain these sort of projects over the 10 or 15 years that you really need to. Okay, so what I did here was just describe what you need. You need time and organization. And all I've done here was, there's a program where you could age yourself by 20 years to see how you're going to look in 20 years' time. So this is not a very good picture of me, probably last year, and this is me 20 years from now. And that's sort of how long you really need in order to do one of these projects from beginning to end, to do it efficiently. And for me personally, of course, I don't have 20 years left in my research career. Hopefully I have 20 years on Earth, but none of my research career. And the reason for this is a really simple one, and that is this gene discovery doesn't take all that long. But the gene validation by a transgenic approach, you get one of three things. This is pretty obvious. You get the trait enhanced. That happens very, very occasionally. You have to do lots of further field tests if this is the case. You get no change where you just throw it out. So that's most of the stuff. It's at least 90% that's no change, or change that doesn't look as good as the wild type. Most of the time you have this sort of effect, the trait partially enhanced, a negative pleiotropic effect. And so at that point, you need to almost always build around it. It's like a pharmaceutical industry looking for new drugs. They find a drug that looks good, and then they are a chemistry that looks good, and they design every single possible chemical compound around it, but it doesn't necessarily take that long to do that. Here, in order to do this, each turn of the circle can take 3 to 5 years. So it's a very, very long and slow process. And that's really what I wanted to say. I just want to finish with a couple things. First of all, I come from the University of Guelph. Most people don't have any idea where that is. This is the Agricultural College main building. We're about an hour from the city of Toronto, which is here, and as I mentioned, we're about an hour from Niagara Falls. And this work was done by a large number of people, both in my lab and a whole bunch of people at Syngenta as well. And I'd be happy to take any questions or comments. Thank you very much.
So thanks, Steven. So I have one comment and one question.
Sure.
So the data you showed from the Nature Biotech paper, where you show very little difference with this rootworm and the BT gene. So I assume that might be from the normal conditions, not under stress conditions. I mean.
So which gene was you talking about? Sorry.
No. The data you showed from the paper, from Nature Biotech paper. She et al.
Oh, she et al. I am sorry. All right, now I got it.
Yeah, so obviously that looks under normal condition, because in normal condition you might not see the difference. You will see the difference only under when there is a pressure.
I'm not defending their data in that paper.
Okay.
I'm just using that as an example, because you can't actually find that's the only paper I've ever been able to find where they did that sort of pretty extensive work. There's all kinds of problems with that paper, but go ahead, sorry.
So the question is about your this GETA 12 where you see the high density, you see better yield. So do you see any difference in the plant architecture, leaf angle, those kind of traits?
Well, okay, probably not for leaf angle, but clearly there's a change in the branching characteristics of the plant. And I am going to forget completely whether that carried down to the roots as well. My guess is it does. And so, you know, you have a much thinner plant, and it sort of makes sense that you can grow it at higher density. But it's not at all clear that if you did that out in the field, it would give the same positive response that we're seeing. So I don't want to claim beyond what I just showed. But I just presented as a, that, you have to think about some of those things. If you see that sort of change in phenotype, it isn't necessarily always going to be bad. I don't know if that makes sense. I wish we could test them out in the field, but we're just simply not allowed. Even the rice lines we're not allowed to do anywhere. So we'd have to remake all of them, and I'm at the point where I'm not going to be doing that.
Yeah, Chem China bought Syngenta today.
I'm sorry?
Chem China?
Yeah, I'm hoping Chem China. I'll tell you the reason. I didn't mention it in the talk, but the OS, the ENOD gene, we got stuck. And the reason we got stuck was that what Syngenta had done was to make the decision to do all of the earlier stage research in their China facility. So they stopped making any corn transgenics in North Carolina that weren't focused on something that had already been largely validated, right? And so the ones that had already been done there, we could test further on, but that one had not been done at that point. So they did it in China. They saw some interesting results in rice, and I don't even know whether they transformed into corn. I assume they did, but they were not, because of various political factors, they were not allowed to do any field tests in China. And so they did all the stuff, but they were stuck even worse than us because their greenhouse facilities were not. At least we have good greenhouse facilities, right? And so it just died, right, for that reason. And now, maybe now that they've been bought by Chem China, now maybe they'll get the right to do field studies. We'll see. One can hope. Yes?
Thank you for sharing all your experience with making transgenic plants. And then I was particularly impressed with the last slide which you put in, wherein you shared your experience saying that the genetic gain through new technologies is not easy.
Well, I think that's pretty clear.
It takes more expensive, lot of cost, lot of time, and all that, as good as the conventional genetic gain enhancement. That's one thing. And then the question I had is regarding your GNC transgenic corn. Right? You mentioned that it had delayed senescence.
That's right. It stayed green longer.
Yeah. I just wanted to know, does it had any implications on the source-sink relation in terms of accumulating higher translocates to the sink?
Okay, so that's actually it's difficult to know. So what I can say is that certainly during the vegetative stage, it does seem to enhance. You get an increase in biomass formation. Okay, at the point of it being transferred to the grain, it's not clear to me. So the initial experiments that were done, so the way, sorry, I should start again. I apologize. So the way the experiments were done, in the first year, the one I showed, that was done using just one hybrid line on a very, very small number of field plots, okay? And it was done in a way that you wouldn't get really statistically valid results of yield in any way, okay? So I can't say anything about the partitioning into the grain, whether it was as effective or worse than wild type, or what the final yield was. Also with corn, you cannot do greenhouse experiments looking at ear formation in any sort of reproducible fashion. You just can't, because you have to self-pollinate. and the ears, the variability is just enormous. And so unfortunately, it's again one of these really hard things for me sometimes. But just at the point when it was getting interesting, it's sort of the plug was pulled on the whole thing, right? And so I can't really say. But to be fair to them, the chance that any one gene like that would actually end up working is small. So even though we're seeing an interesting change in phenotype, the chance that it would still work all the way through commercial development is small. And so I can't really blame them from a money point of view, but it sort of hurts in here that we couldn't carry it forward. I don't know if that answers your question. We really don't know the answer.
Yeah, getting back to the genetic gain thing, definitely it's expensive, using modern technologies. What's your recipe, when there is no traits, or you have trait barrier, or you can't break it? For many crops we see, including our own crops like pigeon peas and chickpeas, there's a ceiling there. You cannot break it unless you do something different.
I'm not arguing against biotechnology, don't get me wrong. I just think we shouldn't be naive about what it takes to do it. And so, I've near the end of the time that I was working with Syngenta, I almost got them to put $10 million a year into developing the capability, in this case just the corn again, because that's what they're interested in, of being able to develop something akin to a little bit to what you're talked about doing here, which is getting the best ideas from the public sector, allowing them to test them out in the field. They would help supply some of the labor. We wouldn't worry too much about, we'd have a very standard intellectual property, all that sort of garbage sort of thing, and really make a real effort to do it. And when you think about $10 or $20 million a year, I think you could do a really good job of seeing whether it's feasible to do some of these things. The problem is that without that sort of consistent effort, it's really hard, because if you're just doing 2 or 3 genes or doing this or that and that sort of thing, it's hard to find ones that could have an impact. And I'm not saying that it would have an impact, but I think there's a chance that you could do it. So I'm not arguing against it, but we need to do it in a formidable fashion, if you know what I mean. And we're spending, more money is spent sequencing genomes, or we're spent sequencing genomes, than doing that sort of thing, right? So it's not a huge amount of money. I used to always compare it to a B-1 bomber in the U.S., $400 million, or maybe it's $1 billion for one airplane. For that amount of money, you could test a crop well for 10, 15 years, right? So it's not a ridiculous amount of money, but anyways, it's a hard sell sometimes. Yes.
Two things. One, your example of the benefits of first-generation traits, and using corn as an example is probably problematic, because you go to other examples like the cotton industry in Australia wouldn't survive without BT cotton, and that's the difference between zero production because of resistance and a flawed-leading industry. So there's big impacts of those first gen.
Oh no, I'm not arguing against them. Yeah. I was just trying to, you have to be realistic about what the effect is on each case, right?
Well, that's plays on my question. I think you answered in the last couple. Is that listening to you, I feel there's somewhat a sober view of the contribution or the investment in transgenics for abiotic or yield improving traits. Is that a conclusion? I'm not in this field, but that's what the impression I'm getting.
I tried to answer it in the last question. First of all, let me go back to that.
But can you go beyond corn then, rather than to
right?
There's a whole range of agricultural products out there, not just corn.
Let me answer three comments about that. Comment one is that She et al. Paper, I don't necessarily believe the final results. I was using that as an example. I should maybe not use it anymore. But I use that as an example because it's the only one out there. There were lots of problems with that paper because of the way they did. The data was over a whole range of germplasm. Who knows what it was they were actually comparing to each other. So I don't want to make too much of that. But it was just to try and demonstrate what you need to do to to see certain types of yield increases in the effort you need to make. That was the point I was trying to make, and maybe I shouldn't emphasize. I should get rid of those bar graphs there and de-emphasize that. The BT and corn rootworm traits are very, very popular. It's not that in corn. It's not that farmers hesitate to plant those, and there's lots of reasons for that. Not least because it acts as a really true good insurance policy under bad insect years. But unlike cotton, corn you can do without, right? So it's not that it's absolutely one or the other. And to your final point on the second generation things, the more multi-factorial traits, to your point, corn is the most bred plant in the world by far. And there's no question that there's also a fair diversity of genetics within that germplasm. And so as a consequence, I think doing this sort of approach on some of the other crops like the ones you use is much, much more likely to see success than with corn. I would say that. But again, the point I would make is if you're going to take that approach, take it. You know, you need to do it in a significant way. Very difficult to do if you're not willing to put the resources behind it. Yes.
Well, it's a question, it's a comment that follows on the others. I mean, I think I like your presentation because in the end, it's as you say, it's a sober view on the use of transgenics to address very complex issues. And I think there's a couple of comments to make there. I mean, the difference between the BT and the Roundup Ready is essentially there are things which do not touch at the productivity of the production sort of framework of a plant. You just put a protein there that kills a bug or that degrades an herbicide, so it doesn't touch on the basics of it. No? And in fact, I mean, what I see from your data is probably once again, I mean, we try to touch on the very complex things, I mean, nitrogen, and you end up with probably what Rajiv was commenting. I mean, you probably change things around the chlorophyll and light interception. So you shoot at something, and when you target something, you get something else. So Monsanto, I mean, the drought, how they call it, drought guard. You know, I mean, they started with what hypothesis? Who knows? I mean, it was really like a shot in the dark, and they end up with a slight reduction in leaf area and disease. That's what drought guards does. So I guess all that comes to the point that, well, any effort with transgenics has faced the problem of basically scaling up. I mean, what may be true at the cell level, I mean, not be true at the organ level, and then you bang into that complexity. And so I'm not saying that transgenic are useless to address complex issues, but I think they would have been more successful when all that started 20 or so years ago, if there had been a bit more consideration of that higher level of plant organization to really target a transformation.
It's not that we don't think about it and have thought about it, through the years, but unfortunately, we're still not at the point where we can say we're going to make a mutation or change the expression of a particular gene, and it's going to have X effect, right? We're not at that point, not close. And I could show you, so typically, I have a number of other examples where you change the expression of a gene, like, not thinking perfect example of this, and you wouldn't think that it would have huge numbers of changes in the rest of the transcriptome, for example. And it's surprising just how much each change has on the change of the expression of other genes. And I think if you did a model of that, one would be incredibly surprised at that complexity. And the problem is that we're also not very good at, so of course, what do you do in this sort of experiment? You take a ubiquitin promoter, right? And that's the first thing you do, right? So you have a choice. Do you lose lots of promoters and fewer genes, or one promoter and more genes? And usually what you do is use one promoter and more genes. And I think that's a reasonable approach. But it's like, banging a thing with a sledgehammer, right? And so in many cases, that was the wrong choice. But the idea really, when we got into this, was, okay, you do that first, you see if there's an interesting change in phenotype, and then you go back and you be more sophisticated. The problem is that going back and being more sophisticated, you have to have the ability to keep doing that, right? And so, like I said, that's 5 years later. You go back and are more sophisticated, but boy, it's hard to keep the interest. So.
Transgenic technology and its history. Thank you very much.
Okay, thanks.